In this lecture, we are going to cover the different medicines that are used for the treatment of type 2 diabetes. And as you'll see on the next slide, the focus is going to be on the medicines that are approved for use in the UK, which, however, um, almost all of them are also in use all over the world as well. So these are basically the main diabetes medicines in general, not just in the UK. And there are a variety of diabetes medicines that are currently in use. And you'll see that as we go through the lecture, they were discovered in many different ways. So you will see examples of medicines that originated from herbal remedies used in traditional medicine. You'll see examples of natural products that are being used, also examples of hormones that have been adapted, for example, to be used as drugs. And there's also cases where the drug has been discovered by accident and not through a rational drug discovery process. And there are also at the other extreme examples of compounds that were discovered using structure-based models using X-ray crystallography. And in terms of their mechanism of action, in how they affect their molecular target, again, you'll see various types of modalities with these drugs. So there are drugs that we will go through that in fact don't have a single mechanism and have multiple physiological effects in cells that then thereby affect the progression of the disease. But there are also drugs that we will go through that have one single target to which they bind with high affinity, and you'll see examples of diabetes drugs that work by binding to an ion channel. You'll also see examples of drugs that work by binding to an enzyme and inhibiting their activity. And in this context, we'll see both reversible and irreversible enzyme inhibitors. And then there are compounds that we will see that bind to receptors and also compounds that affect the biological activity of transporters too. And in the end, the clinicians need to make a kind of a complex decision on which drug to prescribe for which patient, and this is done by various criteria. First and foremost, it will depend on how effective the drug is in treating the patient and lowering their glucose levels. Then there are also drugs that come accompanied with certain side effects, and it will depend on whether those side effects are important for that particular patient or not. So, for example, some of these drugs almost inevitably have a potential for weight gain. And since obesity is kind of a precipitating factor for diabetes, so this can be a double whammy that should be avoided if possible. And then there are some of these drugs, as we'll see, in terms of the way they act pharmacologically, they do depend on the secretion of insulin by the pancreatic beta cells. And if the patient is deficient in insulin production, then these drugs will not have the maximum benefit to them. Some of these patients will be taking other medications. So then there's also the question of a drug-drug interaction that might prevent certain combinations from being co-administered. And last but not least, these drugs vary quite a bit in their costs. There are some drugs that are quite cheap to make and also relatively ancient, so they are not under patent protection, they are generic, and these are often used as first-line therapy. And that there are some of these drugs which are quite expensive, and then the decision to use those drugs will depend on the patient's needs. And in this slide, you can see a kind of a chart of the current and future situation with diabetes. You can see that it affects a significant number of people around the world. But furthermore, the projections worldwide are that diabetes will continue to be on the rise. And you can see in some parts of the world, there's a predicted to be a very large increase in the incidence of the disease over the next 20 years. So, for example, as a whole in the world, the prediction is that there'll be a nearly 50% increase in the patient size, but then you can see in certain areas this is exacerbated. Like in Africa, you're seeing more like 150% increase in incidence, and similarly in Southeast Asia, over 80% increase in incidence. And in general, people of Indian descent, like myself, have a higher susceptibility to diabetes for genetic reasons. And the treatment of type 2 diabetes is kind of summarized here. Firstly, the patient is diagnosed to have diabetes, and initially this is usually done by performing tests to identify the level of glycosylation of hemoglobin. And hemoglobin is the oxygen carrying protein in the red blood cells, and hemoglobin over time will become glycosylated, and the longer the lifetime of the protein and the higher the amount of glucose that's present in the blood, the higher the level of glycosylation of this protein. And if you measure the level of glycosylation of hemoglobin, that then gives you indirectly a measure of how much glucose was present in the bloodstream over the last 60 days. And while 60 days, that's because that's the rough half-life of a human red blood cell. So tracking the glycosylation level gives you a decent idea of glucose levels in that individual. You can then further confirm this by carrying out what we call a glucose tolerance test, where you give the person a significant amount of glucose and then see how long it takes the body to clear this level of glucose, and that will give you an idea of whether the person's metabolism of glucose is normal or not. And if the person is diagnosed with diabetes, then there are various options. In the first instance, it might be possible to correct by changes in the lifestyle. So changing the diet and eating more healthy foods might help. If they can bring down their weight and increase their physical activity, that will also be beneficial. If this is not sufficient to bring down the levels to a reasonable level, then we have to resort to medication. And as I said, there's quite a few diabetes drugs, so many different treatment options. And first-line therapy around the world is monotherapy with metformin, and we'll cover what this drug does in more detail in a minute. And if that's not working well enough, then you can have monotherapy with other classes of antidiabetic drugs, like the sulfonylureas or other drugs that are administered orally. And if the monotherapy doesn't work sufficiently, then there's the option of using a combination of two or more drugs that work by different mechanisms. And in addition, if necessary, the patient can be injecting insulin as the need arises. And on this slide, I show you a complex chart, if you like, of how these treatment options are decided in the US. And the reason I've picked the US is that the US spends far more per individual on healthcare than any other country in the world. And you can see that even in that situation, one of the major drivers for the prescription options is cost. So in the US, patients, if they are fortunate enough, will have private insurance that pays for the cost of these medicines. But even so, there are some medicines that are a lot cheaper than others. And in some cases, the option will be decided on cost rather than the efficacy. So with that short introduction, we'll go through the first of these drugs, which is metformin. And this is a drug that's really inspired by herbal traditional medicine. as you'll see in this next slide. So the origin of metformin comes from the fact that the plant, the lilac plant, Galega officinalis, was used for the treatment of one of the side effects in diabetes, and that is the painful and frequent urination. So this crude extract had been used traditionally, and later on the pure compound that was responsible for this pharmacological activity was isolated, and that was found to be this guanidine compound galegin. So this is a very simple looking molecule. It just has one functional group, a guanidine, and an alkyl chain of 5 carbons attached to it. And as a pure compound, it was in fact briefly used for the treatment of diabetes. However, it had issues with toxicity, and for that reason was discontinued. So then medicinal chemists were interested in making analogs of galegin to see if they could come up with more effective compounds. And medicinal chemistry usually progresses by quite simple ideas and concepts. And one of these that turned out to be quite successful in the context of metformin was the fact that the molecule only contains one functional group, the guanidine. And the idea was that if one guanidine is good for the drug-like properties of this molecule, then maybe two guanidines is even better. So then synthetically, medicinal chemists started making a variety of compounds that contained two guanidines within them, connected by a spacer of varying lengths. And one example shown here, this is syntalin, so this was also used clinically, but also withdrawn due to toxicity. And then people started experimenting with the distance between these two guanidine groups. In syntalin itself, you can see a long chain separating the two guanidines. And later on, people tried different variants of this. And it turned out that actually the best compounds for clinical use are those in which there's no separation between the two guanidine groups. And in fact, all you need is to link the two guanidines together. And that led to what we call now the biguanides. And there's 3 examples of these shown here. On the left, you see fenformin. So this has this double guanidine unit, the biguanide, and attached to it is 2 carbons plus a phenyl ring. And then there's also buformin, which has the again the biguanide unit, and now a spacer of 4 carbons. And these two were also used for a certain length of time, but they had this side effect of lactic acidosis, so they were withdrawn. And from this class of molecules, the one that has withstood this test of time is metformin. And in metformin, you'll see the carbons attached to the biguanidine unit have been also stripped away to the bare minimum. So in metformin, all you see is 2 methyl groups of 1 carbon each. And metformin had the best overall profile of efficacy and reduced toxicity and side effects. And that became one of the early drugs that was approved for the use in diabetes about 60 years ago. And this is one of the drugs that is in the WHO list of essential medicines. And this is a list compiled by the WHO of medicines that are considered to provide the best healthcare benefit around the world in terms of, firstly, their efficacy and providing benefit for patients, but also in terms of their acceptable side effect profile and toxicity. And thirdly, also in terms of cost. So these are all drugs that are intended to be safe and effective, and at the same time affordable, and not just restricted to use in more affluent Western countries. And metformin is cheap because it can be made very easily. So at the top of the slide, this is really an organic synthesis point, so it's not really necessary to know for this lecture, but just as an aside, you can make metformin by just reacting cyanamide So cyanamide has a amine with a lone pair on it, which is nucleophilic, and this can then react with the second molecule of cyanamide to give you this dimer. And within this dimer, you have this CN group, which is electron withdrawing. That reacts with another nucleophile, which is dimethylamine, which has its lone pair. That comes in and attacks the nitrile, and that gives you metformin. So you can see that the synthesis itself is very simple from very simple starting materials, and that means that metformin can be made on a very large scale at a relatively low cost. And throughout this lecture, we'll be also covering some of the physical chemical properties of these drugs as we go along and try to understand their properties as drugs and how they relate to their use. And you can see metformin is actually a very unusual drug compared to the typical drugs that you encounter. And you'll see that it has very few number of carbons. There's only 4 carbons in metformin. And to give you a reference context, the ethanol, which is not a drug, but we often consume and drink, maybe even in excess, ethanol has 2 carbons, right? So metformin only has 2 more carbons than ethanol. And meanwhile, you can see it even has more heteroatoms than carbon, so 5 nitrogens compared to 4 carbons. And that again is very unusual in an organic drug. Normally you see lots of carbons and fewer nitrogens and oxygens. So to be honest, to medicinal chemists, this is the kind of drug that is a bit weird in structure. It looks too simple to be effective and doesn't look like the kind of molecule that would bind with high affinity to some molecular target and give you a significant physiological response, but in reality it does. And in fact, it is the first line therapy for an important human disease. And to go along with this small size, you can see that the molecular weight of metformin is very low, again, compared to many other drugs. It has a very low lipophilicity because, as you can see, it has very few carbons. It's mainly crawling with nitrogen atoms, which are polar. So this is a drug with a negative log P, i.e. it is more soluble in water than in octanol, so it is highly solubilized in the aqueous environment within the body. And in terms of other kind of physical chemical properties, it has 5 hydrogen bond acceptors, 4 hydrogen bond donors. It doesn't have any rotatable bonds. It has a relatively low surface area. So all these parameters suggest that this is a drug that can be administered orally. The only one that we would question is this log P to enter the cell. This is a molecule that needs to be absorbed through the lipophilic cell membrane, and that is not going to be likely with such a negative log P, which keeps this in the aqueous environment. So you can have drugs with very low log Ps, but either they are working on targets that are on the surface of the cell, so that the drug doesn't actually need to get through the cell membrane, or they are operating through some other mechanism of absorption. And that is the case in metformin itself. So metformin has very poor passive penetration of the cell, but it is recognized by a transporter in the cell surface, and it's then brought into the cell using active transport. And as long as a cell is transporting a molecule in by active transport, then it doesn't need to follow some of these guidelines for oral bioavailability. And one of the other interesting points about metformin that make it unusual is that this is a drug that we have been using for more than 50 years. And in fact, it is the first line therapy for diabetes, as I've said. Nevertheless, the exact mechanism of action of this drug is not fully understood. So even a very well-known drug like this is still a bit of a mystery at the molecular level. What we do know is that, as I mentioned, it is transported through the cell membrane. So it comes in by being recognized by this solute carrier OCT1, which is a transporter sitting in the cell surface. And it comes into the cell through this process. And once it's there, the main action of metformin is within the mitochondria, which is the organelle in the cell where metabolism takes place. And basically, you can say that metformin is affecting the energy and metabolism in the cell. And by doing that, it then has a positive effect in the treatment of diabetes. And here's in more detail specifically what it does. By inhibiting the mitochondrial complex 1, this is where ATP is being produced from AMP. And by inhibiting this process, metformin results in a reduction of the ATP level, and by doing that, an increase in AMP levels because the AMP is no longer being converted to ATP. It also reduces the NAD plus levels as a result of this inhibition of the complex. And these events then cascade into a number of different pathways, which can then have a beneficial effect in terms of diabetes. So firstly, in the liver, reducing ATP levels leads to a reduction in the production of glucose in the liver. That's the process we call gluconeogenesis. And as you can see in this slide, the reduction in ATP leads to an inhibition of a number of these enzymes that are involved in the gluconeogenesis pathway for glucose production. The AMP that is increased due to the action of metformin, AMP itself then binds to and stimulates a protein called appropriately the AMP activated protein kinase. So this is a kinase that binds to AMP and is then activated, and the downstream effects of that are also helpful in the context of diabetes. Furthermore, the AMP that's produced binds to GPCR. This is the glucagon receptor. It binds to and inhibits the adenylate cyclase that is part of this GPCR and inhibits its activity. And by doing so, it then reduces the level of cyclic AMP in the cell. So there are several different complex pathways by which metformin exerts its effect. And as I said, it's not completely understood how it works. It's a very simple looking compound, but nevertheless very effective. And in terms of using metformin as a drug, as I mentioned, it's a very inexpensive drug. It's also quite well tolerated, and there are clinical doses that are multigrams per day. And this is actually a high level, not only in absolute weight, but also even more so in terms of molar value, because as you remember, metformin is a very small molecular weight drug compared to many other drugs. So the molecular weight is just over 100. So even a gram is equal to 10 millimoles. And there are patients who are taking multiple grams per day. And that's not that common for many other drugs that are used on a daily basis. And the main reason it's a popular drug for the treatment of diabetes is that even though it's an old drug, it has often proven in practice to be more effective at reducing glucose levels than many of the other antidiabetic drugs that are in use. And that's why it has remained first-line therapy. And this is one of those drugs, and you saw in the previous slide the pharmacology of how it acts. And for its action, it does require endogenous insulin to be present. And that means that this is the type of drug that is best used only if the patient has functional pancreatic cells still present that are still producing insulin endogenously. And this is not a drug that causes either hypoglycemia or weight gain. So those are possible side effects with some of the other drugs that we'll see. And it's a relatively safe drug as well. It doesn't have a huge number of side effects associated with it. They are mainly gastrointestinal side effects. And in some rare cases, these guanidine compounds also lead to an increase in acid levels due to lactic acid in muscles and so on. Another nice point about metformin is that it's not really contraindicated with other drugs. So if the patient needs to be put on other oral drugs for the treatment of diabetes, then that's fine. And as I said, it's still a bit of a mysterious drug in the way it works, but we do know that it does significantly affect cell metabolism. And that being the case, there are other conditions in which that can be potentially useful. So, for example, there's a lot of interest in using metformin as an anti-cancer drug. There are clinical trials ongoing to examine metformin because in cancer too, the energy metabolism is seriously disrupted, and it's possible that it might be corrected through metformin. And another popular use, mainly by people who are self-medicating, is the fact that your metabolism changes with age, and it's considered that taking metformin or on a daily basis can help correct for the slowdown in metabolism as a person is aging. So there are people who take metformin daily for this reason, even though it's not prescribed drug for that. The next class of drugs we will look at are the sulfonylureas and meglinitides, and I put them together because they both work on the same molecular target. And this is an example of drug discovery where these drugs were discovered in an accidental way based on side effects that were observed in the clinic. And you can see here where the story started. And these were sulfonamide drugs. These are belonging to the sulfur class of antibiotics that were quite popular in those days, but have been supplemented by other antibiotics like penicillin nowadays. And in this particular compounds shown here, there were some clinical trials carried out in the 1940s and later in the 1950s as well, where there were side effects and even deaths observed in the clinical trials. And these were attributed to serious drops in glucose levels, i.e. hypoglycemia. And in a normal person, this would be dangerous, but if the person is diabetic, then lowering their glucose level would actually be quite useful. And this eventually led to the clinical drug shown at the bottom of the slide. This is tolbutamide, and this compound had this hypoglycemic effect of lowering glucose levels, and it did not have the antibiotic effect. And this was also approved by the US FDA about 60 years ago. And it's a short-acting compound because it undergoes drug metabolism quite rapidly by oxidation of this methyl group on the aromatic ring. And there are some potential concerns with this compound over possible cardiovascular side effects, and you'll see later on there's newer compounds in this class that tend to be used. And for a long time, these sulfonylurea drugs, as they were called, were of unknown mechanism pharmacologically, but in more recent times, their actual molecular target was identified. And it turns out that these sulfonylurea drugs bind to a particular ion channel, and this is a potassium channel called the potassium ATP channel. And these are channels that are made up of heterooctamers. They are made of 4 subunits called the Kerr 6.2 subunits, which are the channel for the potassium conduction, and also for regulatory subunits called the SIR1 subunits. And these are very large proteins, ion channels, and they traverse the cell membrane, and in their open or closed state, they can then lead to the transport of ions through the cell membrane. And if you look at the left side of the cell of the slide, you can see what the channel looks like in its resting stage in a pancreatic cell and the consequences of that. So in the resting state, when you have low glucose present in the surrounding medium. So this would be, for example, in a fasting state or before having a meal. The glucose is recognized by the glucose transporter, and then that is then taken up by the mitochondria, where it's then metabolized. And when that happens, you'll get ATP produced. But in the resting state, the cell has relatively low levels of ATP and higher levels of the ADP. And that then leads to the ADP binding to the channel, and this keeps the channel open. And when this potassium channel is open, potassium will then exit through this channel from inside the cell to the outside, and that leads to a change in the membrane potential of the cell, and that keeps the cell membrane at a hyperpolarized level. And at that hyperpolarized environment, that keeps this other channel closed. This is a calcium channel which responds to voltage, and when the voltage is hyperpolarized, that keeps the calcium channel closed. So that's the resting state environment of the pancreatic cell. Let's move on to the middle of the slide, where you see what happens when there's a meal or there's an influx of glucose. So when there's high glucose levels, this is taken up by the glucose transporter, and this then gets into the mitochondria, and that leads to metabolism and the production of ATP. And with the production of ATP, that keeps the potassium ATP channel closed. And when the channel is closed, the cell membrane is depolarized, and this in turn leads to the voltage-gated calcium channel being opened. And when this calcium channel is opened, that then leads to the vesicles that are containing insulin to be broken to release their cargo, which is insulin, into the bloodstream. So that then leads to a feedback loop. You are in a situation of high glucose, insulin is then being produced, so that tells you that to basically start consuming this glucose. And what these drugs do is that they bind to this potassium ATP channel and block it. So they kind of give you the same effect as when the channel is closed by ATP. And on the right of this slide, you'll see a kind of cartoon view of the ion channel, and you can see that it can be stimulated by certain molecules, so like ADP, for example, in the physiological context, but it can also be inhibited. Again, physiologically, it's inhibited by ATP, but then it can also be inhibited by drugs, chiefly the sulfonylureas or glinides. And in this cartoon, it also shows you where within the ion channel these drugs are binding. So there are these different sites on the intracellular site of this ion channel to which these drugs bind. And by doing so, they then keep the channel closed, and that leads to the insulin promoting activity of these drugs. And the SAR, if you like, of these types of drugs in terms of designing them is relatively simple. So you have this sulfonylurea functional group. So the name says sulfonylurea. So here's the sulfonyl part, here's the urea, hence the name sulfonylurea. So that functional group is of course important. So you have to have the sulfonylurea. And then it's a question of what you can attach at either end to the sulfonylurea. And on the sulfonyl side of the molecule, you have an aromatic group in these drugs. And the key thing to avoid is that you should not have an amine where R1 is present. And the reason is that if you put an amine in R1, then this is a compound that will also be an antibiotic. Remember, these drugs were discovered from antibiotics due to this additional hypoglycemic activity. So you want to avoid having any antibiotic activity. So in these drugs, you won't see a nitrogen in this position, but some kind of long chain with some substituent attached. And then on the other side of the molecule, R2, in the urea, there can be various substituents attached. And by playing around with the substituents on R1 and R2, different companies have come up with different drugs in this series. And the ones that are approved are shown here. So here are some second generation sulfonylureas, gilbenclamide, which also has the other name glyburide, glycoside, and glipizide. And they are slightly different in the pharmacokinetics. So some of these drugs are relatively metabolized rapidly, especially the ones which have like a methyl group at the aromatic ring, which can be oxidized. So those tend to be more short acting. And then there are other compounds that are more long acting, like glibenclamide. And then here's a third generation sulfonylurea, which is another example of a long acting sulfonylurea, which doesn't have that aromatic methyl group prone to metabolism. And with this compound, glimepiride, you can see that its physicochemical properties are typical of what we expect for an orally bioavailable drug. So this is a compound below molecular weight 500. The log P now is about 3, so this is a drug that should have no issues in being absorbed by passive absorption through the cell membrane. And the other properties are also compliant with the suggested values for oral bioavailability. And as a class, these compounds are again quite inexpensive to make, and they were in fact the first class of oral antibiotic drugs in clinical use since the 1940s, almost 100 years ago. And what are the major side effects? As you may remember, they were discovered accidentally by the side effect of hypoglycemia. And so this is a general attribute of the drugs in this class. They can cause hypoglycemia. And at the same time, they can also cause weight gain as well. And one feature of these drugs is there are several of them in this class, and they vary according to their metabolic stability. So that means you can also fine-tune the drug according to what is most beneficial to the patient, whether it should be a short-acting drug or a medium-acting or long-acting drug. And as we saw in the mechanism of action of these drugs, they work by binding to the potassium ATP channel in the pancreatic cells. So this is another example of an antidiabetic drug that will only be working if the patient has pancreatic islet cells that are functional present and that then secrete insulin. This is a drug that can be used in combination with other antidiabetic drugs, metformin, for example, as well as others, and insulin. And once the kind of mechanism of these sulfonylureas began to be understood, and the fact that they bind to this potassium ATP channels, then there was a lot of interest in trying to discover other compounds that are not sulfonylureas in their chemical structure, but can have the same effect of binding to these channels and thereby have the same medicinal value. And there's two now that are in use and clinically approved that are shown on this slide. And these compounds are compared to the sulfonylureas, they do not bind as tightly to the ion channel, but for the same reason, they also dissociate more rapidly too. So these are short-acting drugs compared to some of the sulfonylureas. And again, there might be patients for which that is superior to having a long-acting drug. And because they work on the same target, they have the same side effect profile as the sulfonylureas, but these effects are somewhat reduced because these drugs don't bind as tightly to the target. And here are the two examples, and you'll see again in terms of their physicochemical properties, they're quite similar and orally bioavailable drugs, molecular weights below 500. Their log P is a little bit on the high side perhaps, especially with repaglinide, but still all right for oral absorption. And repaglinide on the left is a drug that came from Boehringer Ingelheim and later licensed to Novo Nordisk, who are one of the companies that specialize in diabetic therapeutics. And a slightly different structure came it's shown on the structure on the right. Interesting compound because it's a fairly simple compound as well. And within it, you can see embedded the structure of the natural amino acid phenylalanine. So this is basically an amide of phenylalanine and this other terpenoid type carboxylic acid. So two very simple compounds that are being joined together as an amide and or can be made in literally like 1 or 2 steps. And you can see its origin is also interesting. This came from Ajinomoto, which is not really a pharmaceutical company, but a food product company, probably most famous for manufacturing MSG, monosodium glutamate. So I'm not sure how they came about this discovery. Maybe they were investigating compounds of this type as food flavoring agents or functional additives to foods. But it turned out to be a pretty good binder to the potassium ATP channels and has since been licensed to Novartis. And with that, I'll stop this first part of the lecture and we'll then move on to some other antidiabetic drugs with different mechanisms of actions.